So what I want to do now, so today we're talking about the issue of if I'm looking at just phenotypes over time, a fossil record, divergence data, some genomic character like expression, or maybe it's some metabolite expression level. How can I use that information to ask if that pattern is under selection or not? And to do that, we have to introduce our first topic, and that's quantitative genetics. So here is your half-an-hour introduction into quantitative genetics. A rule I always give to let people, how do you assess when you know something? There's a very simple rule. I call it the grandmother or grandfather test. So you've just had this lecture, and you call your grandparents, and they ask you, what did you learn today? Can you tell them in 2 or 3 sentences? And the reason I frame it in terms of your grandparents is you're not going to use any technical jargon. You're not going to say we learned about the eigenstructure of the G matrix. You're going to basically tell them in plain English. And the reason it's a good test is that all the stuff we're going to talk about has details. But the details often confuses the basic question. The way to learn complex material is to focus on the basic question, and the details follow from that. Students typically get fixated on the details and forget the basic question. The details are stuff you look up. That's why we wrote the big textbook, to look those stuff in. So in quantitative genetics, the basic question is, how do you follow complex phenotypes and determine how much the variation is genetic and how much is environmental? And the key is you look at sets of relatives. Sets of relatives share genes. If you randomize sets of relatives over environments, you randomize the environment, and therefore you can separate effects from shared genes from effects in shared environments. Now, there's situations where you can't do that, like humans, where it gets more interesting. That's the basic idea behind quantitative genetics. So here we go. Here's what we're going to cover. Fisher's variance decomposition, this critical concept called additive genetic variation and heritability. We'll talk about inbreeding and effective population size. We'll talk about when you have a quantitative character, mutation is how much new variation in the trait is introduced by mutation each generation. We'll talk about additive variance under a balance between drift and mutation. Then we'll finish by talking about the breeder's equation and selection response. This is kind of all background we're going to use then to talk about whether an observed pattern is too extreme relative to drift. Now again, I want to stress at the outset that there's a lot of material here. And so if I slip in a concept by mistake you hadn't seen before, do not hesitate to ask me to expand on that, right? And again, the other thing is, this course is like a gigantic fire hose of information, just shooting all this information at you. So you don't want to necessarily try to grab everything. That's what the notes are for. You want to kind of sit back and get an overview of the basic ideas. Then you can go back and read the notes if you need more details. If you really need more details, you can go back and read the book. By the way, I hate continue saying read the book because it sounds like I'm trying to, sell copies. I get about $5 a copy. I'm not going to make any money by doing that. But what you need to know basically is number one, be introduced to a concept. If that concept's in your mind, then when you need it, you then know where to go look up information about that. First place is the notes. And by the way, what we'll do is I always find typos in the notes. This is the first time I've taught this course. There'll clearly be typos. So what I'll do is I'll upgrade and adjust the notes after each lecture. But be sure at some point and download all the notes. And think about this. Hopefully this material will be using 5 or 10 years from now. So you want to think now about how am I going to download the notes in such a way that I can go back and use that material. Probably make some nice file structure, maybe put some additional notes in a Word file or something on that. But think now about how you're going to access this information 5 or 10 years from now. So if you think about it now, it's easy to do. 5 or 10 years from now, it's like, where did I put that? Oh my God, it was on a CD. What the hell's that, I mean, I actually have these weird things called paper notes, and they have these little weird sticky pieces of paper on them that index interesting things. That's kind of really old school. So basically, there are two books that get you into quantitative genetics. Here's the classic by Doug Faulkner, and Mike Lynch and I put this together 20 years ago. By the way, we call this Volume 1, and just to remind you, to give you some idea here, we want to emphasize that it applies to a whole bunch of traits. That's Mike Lynch's daughter there. His son is actually standing in the cornfield. You can't see it. No one can see it. So we call that Volume 1. That's basically the background in quantitative genetics. We say that because Volume 2, which is not Lynch and Walsh, but Walsh and Lynch, which is coming out, asks about the evolution and selection of the traits. By the way, a couple of fun facts on here. That's my wife's little rabbit when he was young. That is Lithopane lei, which I actually named a moth named after my wife. So bright pink thing. So Mike got his daughter and his son. I have my wife's moth and her rabbit on the cover here. Just bizarre, but so Faulkner is what I learned basic quantitative genetics from. Faulkner is extremely nice because basically it's accessible. If you want to pick up quantitative genetics from scratch, Faulkner is now Faulkner and McKay, Trudy McKay, updated a little bit. It's kind of the standard introduction. If you want to know, so it's about 400 pages. We're about 1,000 pages. If you want to know where all the bodies are buried, where all the problems come in, then you go to Lynch and Walsh. So if you want to get into quantitative genetics, Faulkner is a good introduction. If you're going to use it extensively, read Faulkner first, then have us around to look at information on here. So everything we'll talk about you can find in detail in both these books. And here's the basic idea. Quantitative genetics traces back to R.A. Fisher in 1918, wrote a groundbreaking paper. This paper was so groundbreaking that the term, you've all heard the term variance before, right? The term variance was introduced in Fisher 1918. It had not been used before. You've all, or most of you probably used ANOVA before, right? The analysis of variance. Analysis of variance was in Fisher's 1918 paper. So what happens in a landmark paper that fuses and solves a massive debate about whether quantitative traits could be Mendelian, and which gives you fundamental concepts in statistics? Such a paper is, of course, rejected. This paper, Fisher's 1918 paper, was rejected by the Royal Society of London, so he had to publish it in the backwater Royal Society of Edinburgh. And he published it with the help of actually one of Charles Darwin's sons. And in this paper, Fisher basically laid out the entire field of quantitative genetics. And he wrote this paper while I was a high school teacher. So the basic idea is, if you look around the room, I always tell people don't look too hard because then HR will come in, there's all sorts of problems. But you'll notice there's a variation in phenotypes. So for example, amount of hair, ear size, which you can see because they don't have a lot of hair, that's a pleiotropic feature. So we observe a phenotype. We often also, in ecology, Z is used quite a bit for a measure of a phenotype. We can break that phenotype into two components. These are what are called latent variables, things we don't observe. So your phenotype is result, number one, of some genotypic value, and number two, some environmental value. And the idea is, if I could clone you and randomly scatter your genotypes over the collection, the so-called universe of environments, if I could do that, that would give me then your average genotypic value. So in theory, even though we don't see a genotypic value in looking at you, in theory we could figure out what it is by cloning you. This happens all the time in plants. And replicating you over a bunch of environments and asking about the average line value. So here's the basic model. And by this, I use different fonts here. This E here, this funky E, stands for expectation or average value. This E here stands for environmental noise. You can also have interaction terms. I won't say much about that. A G by E term simply means that you can have scenarios where you might have the rankings of the two genotypes in environment one are this, for example, a wet year, or it might be insects feeding on a certain plant. If I go to a dry year or a different plant, it might be that that gap between the average value of the two genotypes now gets smaller or larger, or even flips. That's an example of a genotype by environment interaction. And again, I won't say much more about that, just simply than to introduce it. Again, there's a lot of material here. There's a lot of discussion of this in Lynch and Walsh. And here's kind of a classic example. This is a kind of an odd histogram. And what you want to look at is, let's look at down here. So basically, these are ears of different sizes from 9 to 15 centimeters. Here's the magic number. So this isn't a bell curve. What you see here is the modal number is about 12.5, and you see you've got outliers over here. So the spread is given by down here. This just shows the size. The actual numbers are given down here. So if I have two, very classic paper by East in 1911, I've got line 1 and line 2. These are fully inbred lines. If they're fully inbred lines, all these individuals, all these roughly 55 individuals or so here, have the same genotype. Why do they have variation? That's environmental noise. Over here, again, this is also an inbred line. These also all have the same genotype. All that spread is also environmental noise. If I cross two fully inbred lines, everybody here is a heterozygote. If this is fixture big A, little a, everybody here is a big A, little a. So again, in the F1, everyone has the same genotype. And again, this is all environmental. What happened in the F2? Well, in the F2, what basically happens is you've got environmental variation, then you also have segregation. If I'm a big A, little a heterozygote, then I segregate out little a, big A, little a, big A, big A. So Mendelian segregation generates variation. So down here, I have a typical scenario for quantitative genetics in that the variation I see has both a genetic component, individuals have different genotypes, and it has an environmental component. So this nice example kind of shows you how you can have environmental components and genetic plus environmental components. Now, of course, this is nice and simple because they're inbred lines. In the real world, how do we handle something like this? Like, how do we handle height? So let's take a break. We're talking about the genetics of complex characters because what we're going to do is we're going to look at phenotypes and try to ask what we can say about evolution from how phenotypes change over time. And so Fisher's key idea, number one, was we can break your phenotype into a piece that's due to your intrinsic genetics, that is, it doesn't change from individual to individual, and the unique environment that individual experiences. Well, an obvious question has to do with, I have a genotypic value. How is that transmitted? So if I'm a clone, and a good example of a clone is I'm fully inbred and I self, or a clone could be something like Daphne or an aphid where I make an asexual copy of myself, then my offspring gets the same genotype as the parent, and therefore the offspring G and the parent G are identical. But when you have a situation where you have sex, so one parent and a second parent contribute parts of their G to make an offspring, what part is passed on? And this was Fisher's key idea. And the key idea is when you have offspring generated by crossing, each diploid parent passes on a single allele to an offspring. If you're a tetraploid parent, you pass on 2 alleles. Hexaploid, etc., 3 alleles. So what we have to figure out then is a parent doesn't pass on all its G, or even half its G. It passes on this special thing that determines the effect of individual single alleles. And so let's figure out what that is. Let's imagine I've got a locus with alleles big A and little a, and here are their average phenotypes. We usually use this, C as just some background value. 2A is the difference between the two homozygotes, and if the heterozygote is exactly in the middle, D is 0, so D is a measure of dominance. This is just simply a way of basically asking what are the average genotypic values. I can then break them into a difference between the homozygotes, which is 2A, and the value of the heterozygote, which is different from D unless it's right in the middle. Okay? So D is non-zero. So if D is positive, it means the heterozygote is close to the larger value homozygote. If D is negative, it's close to the smaller value. If D is 0, it's right in the middle. So the key concept in terms of what a parent passes on to its offspring is this concept called the average effect of an allele. And let's imagine the following. So here I have this lovely obesity allele, this Pepsi allele, and the average effect is simply this. You're a random offspring and you have a copy of this allele. And the other allele is picked at random from the entire population. What is your average value, given you've got this allele and the other allele is random? The other allele could be a second one here, but it's picked at random. Right away, it tells you a couple things. Number one, the average effect of an allele isn't just a feature of the allele itself, it depends upon the population. Because if I have a population and everyone here is the competing Coca-Cola allele, then the average value of this is going to be quite different from a population where this allele isn't there. This allele is still the same, but remember, it's the average effect of this allele and a random allele from the entire population. So the average effect of an allele is population specific because it depends upon the population you're drawing it from. So we call the average effect of allele A the mean offspring value given that a parent transmits allele big A and the other allele is random minus the overall mean. So for example, if the allelic effect, let's say in this population, let's say the population is, let's say Brazil, the allelic effect of this allele could be 3, which means that if you get this allele and the other allele comes from a sample from a Brazilian population, you're 3 units above the mean. If I go, for example, to, let's say, a Florida population, the average effect of this allele might be -1. It means if you get this allele and an average allele from the Florida population, you're 1 unit below the population mean. So the key thing is when I'm a parent, when I pass on my genotype, here are my 2 alleles, what I do is I give 1 allele to the offspring. That's the part I pass on. So I only pass on what the average effects of that allele are. Now that's kind of a deep concept. So there's something I modestly call the Walsh curve, which helps you understand these. And this is time, and this is some measure of understanding. So at time 0, you've never heard it before, then I mention, that makes sense, right? And then what happens? You think about it, right? And so what you see here is that over time your understanding is increasing, but in certain parts of time your understanding goes down. Why? Because you thought about it. Don't think, if you're going to think really hard so you jump over here. And the key idea in a lot of these concepts is if you just take them at face value, you understand most of them. So the average effect basically is if I transmit an allele to you, how much above or below the population mean does that meet you? Question? No. Okay, just making sure your hat doesn't blow away in the big wind in here. So this is what this is what parents pass on. They don't pass on G. What they do is they take every allele they have, they look at allelic effects, and it's the sum of those that they pass on. And I'll talk about that concept in a second here. So you can actually figure out what that is. Suppose a parent contributes a big A. So if P and Q, this is the other allele, then with probability P, the other allele is a big A, and they're a big A. With probability Q, the other allele is a little a, and they're a heterozygote. So if I change populations, I change these frequencies. I'm either a big A or a big A little a, but these frequencies change. So then for this population, I can compute the mean given a big A and can compute the average effect. So the key here is that my G value, I don't give all that to my offspring. What I give are single alleles. So what I pass on my offspring is something called the average additive genetic value. It's also called the breeding value. And to compute that, I simply look at suppose I have allele I and allele J at a locus in me. Well, the breeding value for that locus, the additive genetic value for the locus, is just the average effective allele I plus the average effective allele J. If I sum over all loci, that gets this thing called the breeding value or an additive genetic value. Now, right now, it's really abstract, and you're probably thinking, how could you ever know that? Because you have to know the underlying loci, which we typically don't know. Then you have to know what their A and D values are. Then you have to know what their allelic effects are. The bottom line is you don't need to know that at all, because the breeding value is something I can estimate quite directly. And here's the key. Suppose I take two parents. So I'm one parent and you're another parent. If you know my breeding value is, let's say, -4, come on, not much luck at, and your breeding value is 10, what do I expect the mean of our offspring to be? Well, the mean of the offspring, then there's a big spread about that, but the mean of the offspring is the average of the two breeding values plus the population mean. So breeding values are deviation from the mean. So if I'm a -4 and you're a 10, add those together, I get 6, divide by 2, I get 3. We expect our offspring to be 3 units above the mean. That concept means you can actually estimate breeding values directly. If I pick a random individual, close my eyes and pick one of you, some are above, some are below, but on average, your breeding value is 0 because it's a deviation from a mean. So if you want to know my breeding value, what you do is you take me and you cross me to a large number of unrelated random individuals. My offspring mean then is my breeding value over 2 plus 0 over 2. So twice the deviation of my offspring from the population mean is my breeding value. So breeding value, even though it has this really abstract notion of where it comes from, you can actually estimate it directly. The key thing to go through this idea here is parents don't pass on G, they pass on the breeding value because they pass on single alleles. Interactions between alleles aren't passed on. Likewise, your allelic effect depends upon the population you're crossing to. So it's not an intrinsic property of the allele, it depends upon the allele and the population you're crossing it to. So breeding values can change. So the key concept here is we actually get something we can actually measure. I can take any individual and estimate their breeding value by doing a controlled mating design. Kind of hard to do in humans, but I can certainly do that in any organism that I can do breeding in. We can actually take that idea and use a procedure called mixed models and do that and actually estimate breeding values by simply knowing sets of relatives. BLUP stands for Best Linear Unbiased Predictors. We won't talk much about that, but much of modern quantitative genetics is basically done by using this basic idea about relatives and extending it up. What BLUP basically does is it has a matrix that takes care of all the pairwise relationships. If you're interested, chapters 19 and 20. But key concept so far is if I'm a parent, what matters isn't my phenotype. Because my phenotype is a predictor of G plus E. If you're a clone of me, what matters is G. If you're produced by asexual crossing, what matters is my breeding value. So I could have a terrible phenotype, but be a really good parent because I've got a good breeding value. Likewise, I could have a really good G value. If you replicate me over multiple environments, you get a high-performing individual. But my G and my A value are basically uncorrelated. So if most of my good value for G is due to interactions, dominance, epistasis, since I pass on single alleles, those combinations aren't passed on, that all gets randomized. Good example, Einstein. Kind of a smart guy. He had average kids in terms of intelligence. Not idiots, but not, Einstein. So Einstein's breeding value for intelligence was very low. His genotypic value, if you imagine it was largely G and not environment, you can argue one way or the other, his breeding value is quite different from his genotypic value. So the key concept, if you want to ask how traits evolve, is this concept of a breeding value. Because if I have a great phenotype, that tells me nothing about my offspring. What I want to have is a great breeding value. And breeding value is simply how much above the offspring, the average mean, are your offspring. It's the sum of the two parental breeding values. So you pick parents with high breeding values, their offspring are going to be exceptional. And that's a concept we're going to come back to. So let me give two more concepts, and we'll review what we've talked about so far. So I can write my genotypic value this way. I have an overall mean. That's the breeding value. So if the alleles are I and J, that's the breeding value. And this is what's left over. Those are the interaction terms. If I have dominance, D is not 0, that term is different. If I have epistasis, non-additive interactions across loci, I get additional terms in there. So I can then write my genetic variation and go through a couple of steps and basically show that my genetic variation equals the additive variation plus everything else. For example, in this case, dominance. So here are the key concepts so far. The key idea is my phenotype is not necessarily a good predictor about what's my offspring going to look like. What that depends upon entirely is my breeding value, not my phenotype. My phenotype is G plus E, so a bad environment may mask a good G. A good environment may mask a bad G. But again, even if I know G, G is not what parents pass on to their offspring unless they have clones. What they pass on is their breeding values. So we have the average effect of an allele. That's a property of a single allele in a particular population. If I keep that allele but change the population, I change the background, I change its average effect. The breeding value is the sum over all loci of the average effects. That is a fraction of G that parents can pass on to the offspring. This is a property of an individual in a particular population. I may have a certain breeding value if I'm randomly crossed an individual in Brazil. I then take the same genotype, randomly crossed an individual in Florida, and my offspring may be different. Why? Because allele frequencies are different in these two populations. So this concept of an additive genetic or breeding value is a property of an individual in a particular population. The variance in breeding values, the additive genetic variance, is a property of a population. This is one of the most critical concepts in quantitative genetics. I have two traits. Trait number 1 has a high additive variance. Trait number 2 has almost no additive variance. If I give those two traits the same amount of selection, which trait is going to evolve? If I've got almost no additive variation, it means all the breeding values are right around 0. So I take two parents and I cross them. The average mean of their offspring is their average breeding values close to 0. Their offspring aren't really exceptional. I could take a really large or a really small parent. If there's very little variation in breeding values, the offspring don't change much. If I have lots of additive variation, a tall individual on average is going to have tall offspring. A short individual on average is going to have short offspring. So a key concept in terms of asking if traits can evolve is their additive variance in an outcrossed population. If the additive variance is big, it means exceptional individuals are probably going to have exceptional offspring. If the additive variance is small, exceptional individuals are probably big because of environmental reasons, not because of that. Good example of this is a colleague I often teach with, Bill Murr, who's a poultry breeder at Purdue. I think someone here was from Purdue. When he first started, he was interested in a trait called shellless eggs. It turns out that chickens at about 1 or 2% lay an egg without a shell. We call it an omelet, right? It's just a membrane around the egg. Now, usually the chicken steps on it, and of course it's gone. The profit margin in a poultry company is 1 or 2%. If you fix shellless eggs, you overnight double their profitability. So there's two explanations. Number one, the nutritionists come in and say they have a lack of calcium. That's why there's no shell. Change the management strategy, change the environment, and you'll solve the problem. Number two, the breeders come in and say, no, it's all genes. If you breed for reduction of shellless eggs, you'll get them. Well, what happened? It turns out that almost all the variation is is genetic. What that means is adding more calcium won't change anything. They're shelled or not shelled, not because of the environment, but because of genes. So changing the husbandry conditions won't change fraction of shellless eggs. The bad news is even though almost all the variation is genetic, almost none of it is additive. So parents that don't have shellless eggs are no more or less likely to have shellless offspring than a parent that does have a shellless egg. So it's the worst scenario. It's almost all genetics. You can't fix the trait by changing the environment, but almost all that is due to interactions. When I give you a single allele, you don't pass on an interaction, you pass on one allele. So in this case, the breeding value is very close to zero, even though most of the variation is genetic. And so if you're worried about how traits will respond to selection, you have to know what fraction of variation is additive. Now, one of Fisher's key ideas is you can actually determine the added variation without knowing any of the genetics. You simply look at how parents and offspring resemble each other. We can use that to estimate the added variation. We can also, by the way, use that to estimate the breeding values directly. So we had all this abstract notation. But the nice thing about quantitative genetics is we don't have to know any of the underlying genetics. None. We can simply look at parents and offspring or known sets of relatives and look at how similar they are and then use that to make predictions about what's going to happen to offspring. That's a key concept. Let me just 2 more slides and we'll see if there are questions. So you can basically, this is another way to write the 3 different genotypes. You can write down an expression for the added variance. What you see is dominance comes into that. You can write down an expression for the dominance variation. If the heterozygote is exactly intermediate, there's no dominance variation. And then the key here is I can estimate the genetic variance for a trait from a set of relatives. And the simplest way to do it is to measure a trait in a parent and measure a trait in the offspring, and that best fitting line, that regression through that, the slope of that regression is given by added variance over 2 divided by the variance of the trait. That's something we call the heritability. So added variation divided by total says what fraction of all the phenotypic variation is due to variation in breeding value. That's called the narrow sense heritability, and that's a key concept. So if you have a trait, one of the key concepts you want to know is what's its heritability. Generally speaking, question a second. Generally speaking, life history traits have low heritability. Egg production, etc., 5% or less. Morphological traits have pretty good heritabilities, 20 to 40%. So to summarize, we'll get to your question. When I'm looking at continuous traits or complex traits where the variation is both genetic and environmental, a key thing we need to know is what is the heritability for that trait? Because that basically tells me how offspring are likely to resemble their parents.
You'll see that equation with the covariance between father and offspring in the numerator there. And I was wondering if you could talk about when these two are equivalent and when they aren't.
So basically, the general expression is there's something called the coefficient of coancestry. And the coefficient of coancestry says if I take an allele from you and an allele from me, the coefficient of coancestry is the probability those alleles are identical by descent. They trace back to a single copy in a common ancestor. So for parent and offspring that are not inbred, this is a quarter, and that gives that covariance as added variation over 2. If I go, for example, to half sibs, I get added variation over 4, If the parents are inbred, I get other things that are involved. So you can make that expression very, very general. It's all based upon this coefficient coancestry. The key point for our purpose, since this is a class in selection, not quantitative genetics, the key part is that what we're basically, if I have sets of known relatives, I know what this is for any trait. Because for all the traits over a set of known relatives, this thing is the same. Then I can simply look at the covariation among those sets of relatives and then compute the heritabilities for any given trait. So there's lots of stuff you could say on here. And we have 200 pages in the book on them. That's the basic idea. Parent-offspring regression, the covariation among known relatives, allows you to estimate the breeding value. That is the added variation. And that's a key concept. Again, key idea, what you tell your grandmother or your grandfather. If you have a trait and the fraction of variation that's due to added variation is high, that trait will respond well to selection. If that fraction, the heritability, is low, that trait will respond very poorly to selection. Yeah?
So this line of reasoning seems to take, so the interaction terms are taking care of epistasis and interactions and dominance into account. Are there any other assumptions about the genetic structure that we need for all of that to work?
Yes. I'm giving you an overview lecture, right? We can drill down in detail into here, but that would then get at some of the other things. So the quick snapshot for now is that I can use resemblance between relatives to look at the added variation. And that's usually sufficient to predict selection response. See chapters 21, 22, and 23 for cases where that's not the case. Oh yeah, no, fine, I'm happy. So what LD does basically is linkage disequilibrium tends to make your added variation smaller. So when you select, my heritability starts out, let's say 40%, it then drops down to 35%. See chapters 16, 17, and 18. Talk about that. But those are good points. I'm not trying to push them off. But for our purposes to move forward, what we simply need to know is a key idea for asking about traits is the added variation they have, or their heritability, which is the added divided by total. Yes? They don't know, because you have to actually find the QTLs and do that sort of stuff. There are mating designs that get at those, but estimating higher variances is tricky. Please. Generally tend to have low heritability. That's correct. That's because you're thinking about evolvabilities, which we'll talk about later. We'll talk about those in length later. Because evolvabilities are related to elasticities, which is an important concept for how you can look at change in traits and related to life history things. So we'll come back and talk about that. Do people know where the term regression comes from? So one person has been well trained. So the original parent-offspring plot was Galton. And Galton wrote a book called Hereditary Genius. And Galton basically took height, adult heights of the average of the two parents, adult height of their offspring, did a plot, and the slope of that regression was 0.6. And Galton looked at that and said, my God, England is going to collapse. They're going to collapse faster than they do in the World Cup, which is pretty bad. And he called this the regression to mediocrity. A slope of less than 1 means what? Got a really good parent over here, the offspring is not quite as good. There's a regression to the mean, regression to mediocrity. Now, of course, he ignored the fact that those really sort of inbred, aristocrats down here have offspring that are more exceptional than they are. So the net result is the population doesn't change. But the term regression comes from the fact that the slope of this regression of heights had a slope less than 1, therefore there's regression to mediocrity, and Galton jumped on this and said, this obviously applies to IQ. It means England's going to get stupider and stupider and stupider. Of course, what it means is the smart one's going to have dumber kids, the dumb one's going to have smarter kids. So it kind of averages out. I like to throw a little history in. As you'll see, I like to put pictures of people who proposed ideas. I think it makes the science a bit more interesting, keeps it a bit more engaged. Okay. Key concept of heritability and additive variation. Now let's move on to something different. Usually you hear drift spoken about in terms of population genetics. We'll talk about it in terms of how it impacts quantitative genetics. And here are two of the people. There's R.A. Fisher, looking very, it's like he just ate an onion or maybe a lemon. And Sewell Wright, I throw this picture in because Wright worked with guinea pigs. And you're all thinking it. You're all thinking it, right? There's the blackboard. He's kind of an absent-minded professor. And that Janice Spofford told me, who was Wright's last student, that you got to get a better ringtone. Come on now. I mean, it's bad when the phone goes off in class. You have a really lame ringtone. It's just really embarrassing. Like Desposito or something like that. Be a bit classier. And she was actually in the class when Wright picked up the guinea pig and started racing the board, because these are big and docile and stuff. So Fisher founded most of modern statistics. Almost everything you see about in basic statistics, except for Bayesian, which Fisher was crazy about, traces back to Fisher. Wright was so English, U.S., University of Chicago, University of Wisconsin, worked for USDA as well. A lot of the key ideas in quantitative genetics trace back to Wright. So the basic model of drift is called the Wright-Fisher process. And here's the idea. So we've got, let's say, 50 people in the room. Means we've got, if we're all diploids, we've got 100 gene copies. I'm going to leave the room so I can't be involved in any lawsuits. We'll lock the door, have an orgy of random mating, and then we'll have exactly as many kids born. So we'll come back a generation later, and there'll be 50 individuals that are a product of this random mating. What's going to happen is, of those 100 genes I had originally, some lineages are going to die off. Some of you, unfortunately, won't get any mates by chance, not because of your phenotype, because it's random mating. Others, lineages will be overexpressed. And so if you run that process over time, eventually all the copies at a given locus will trace back to a single founding copy. Move to a different gene, the same thing happens, but they'll trace back to a different founding copy. That's the basic idea behind drift, and Wright and Fisher were the one that worked out some of the mathematics. So suppose initially in a population of 2n, so n is 50, let's say 100 individuals, I've got 14 copies of an allele. So my frequency is 14 divided by 100. What I want to know is, in the next generation, what's the chance I've got 17 or 24 or 0 copies? So I move from I copies to J copies. The Wright-Fisher model, and there are lots of models, something called the Moran model, for example. The Wright-Fisher model is binomial sampling. There's a typo in here. Everywhere you see n, it should be 2n. I was too lazy to retypeset this, so I'll go back and fix that. Basically, this is binomial sampling. P is the chance of the success. 1 minus P is the chance of a failure. So what's the chance you get out J copies? It's having J successes when there's the frequency of a success. That completely specifies the allele frequency changes. If you run that experiment over time, eventually, as you run it long enough, you're going to get allele is either lost or fixed. Also notice that it depends upon n. So the Wright-Fisher model for drift, when n is large, the new value of P is going to be very close to the old value of P, because if I have 1 million individuals, random sampling isn't going to change that frequency very much in one generation. Over many, many generations it will. If I've got 10 individuals, random sampling could cause them to be lost in one generation or become fixed in one generation. So as your population size gets smaller, the noise from random sampling gets bigger. That is, the time scale for drift to be important gets quicker. So large populations, over a few generations, drift isn't very important. No matter what your population size is, over a sufficiently number of generations, drift is very, very important. So drift causes alleles to become lost from the population. In fact, if my initial frequency is P, then simply under random mating, so alleles have no fitness advantages, the probability the allele is fixed is going to be p, the chance it's going to be lost is 1 minus p. So if the frequency is 17% in a population of size 50 or a population of size 10 billion, in both populations, the one allele is eventually going to become fixed, and the probability in both those population sizes of that allele being fixed is 17%. It's independent of the population size. Where population size matters is the expected time to fixation. That expected time scales with n. So if this room is of size 50, so 4n equals 200, it means on average, after 200 generations of random mating, any given allele with this ignoring mutation can be, every allele here at a given locus will trace back to a single copy in an ancestor. That's one way of looking at drift, and that way of looking at drift is called a coalescent process. We can build it up this way. Suppose I pick a random allele in me and a random allele in you. The chance they have a common ancestor one generation ago, we're going to assume we're all hermaphrodites, doesn't change things very much. Well, it might change your lifestyle, but it doesn't change the probabilities very much. Woody Allen's line about it doubles your chance for a good time on a Saturday night, I think is how he put it. So basically, the chance that the copy in me and the copy in you come from a common ancestor one generation ago is simply 1 over 2n. So if those that know about a geometric distribution, with success probably 1 over 2n, it means on average we have got to go about 2n generations for that allele in you and allele in me to trace back to a common ancestor. For the entire population, so pick a random allele in me, a random allele in you, 2n generations back to a common ancestor. Again, there's a spread about this, an average value. It's got a long tail. What about the entire population? It's only twice that. It's only 4n. So 2 alleles take 2n generations, but the entire population only takes 4n generations. So kind of summarize about drift so far is random change in allele frequencies result in the loss of genetic variation. Allele frequencies change. Eventually they'll change to reach either 0 or 1 without any force introducing new variation. That allele now shows no variation because it's either fixed for one copy or the other. The power that is the time scale of the drift is about 1 over 2n. So for example, if I have very weak selection, when can selection overpower drift? Well, the time scale for selection is about 1 over s, where s is the fitness advantage. So if 4NES on average is less than 1, it means drift overpowers. If 4NES on average is bigger than 1, it means selection overpowers. That sounds like an abstract concept. Let me make it very real. How many people have heard of codon usage bias? So about half. Codon usage bias is that if you look at our genetic code, there are many synonymous codons. A different triplet codes the same amino acid. And in many cases, you've got a scenario where the fourth base position, no matter what it is, all codes for the same amino acid. So you might expect, okay, the frequency of those 4 codons simply match the frequency of ATCG in the population. Right? We don't see that. In bacteria, we see enormous bias in that, for example, if I've got 6 different synonymous codons, one of them is used 80% of the time. If I look in mammals, I don't see that except in highly expressed genes. There's clearly selection, and the idea is what's happening is the codon that has the highest frequency of acceptor tRNAs is the one that's preferred, because therefore it will bind on the ribosome quickest and therefore allow translation to go a little bit quicker and maybe more efficiently. So people have argued, well, the reason you see codon bias in bacteria and not in mammals is because in bacteria, speed of replication is extremely important. The selection coefficients are different in humans and bacteria. A population geneticist could say, no, There's no evidence that S is different in bacteria and humans. The difference is the population sizes are much smaller in mammals. So if S is on the order of 1 in a million, in bacteria, 4 NES is a big number because NE in bacteria is big. In mammals, human effective population size is 50,000. 4 times 50,000 times 1 in a million is a number much less than 1. So drift overpowers in mammals. In bacteria, selection overpowers. Selection coefficient's the same, just the power of drift is different. So your effective population size really matters. And Mike Lynch talked about this at length in his book on evolution of genome architectures. So the power of selection matters. The time for 2 random sequences to have a common ancestor, the coalescent time is 2 N generations. The coalescent time for the entire population is 4 N generations. So we're going to put these features together. So drift causes allele frequencies to randomly change. The speed of which that happens depends upon the population size. I mentioned effective population size. We'll talk about the difference between those in a second. So what we can then ask is heterozygosity. An individual is a heterozygote. Well, if it's initially a certain value age 0, what's its expected value at time t? Why is it going to change? Because as allele frequencies change, heterozygosity is maximized when the 2 alleles have frequency a half. As the alleles drift to loss or fixation, the frequency of heterozygotes goes down. There's a simple expression. Here basically is what it is. That's the heterozygosity at time t, depends upon initial heterozygosity, and this thing here, if you think about what this is, as you make t larger and larger, this thing goes to 0, so heterozygosity goes to 0. So if you have drift, over time, heterozygosity goes to 0, and the rate at which it occurs depends upon the population size. That'll be important. And it's important because of this. Additive variation. Let's go back and look at our expression I went through quickly for additive variation. There it is. 2p1 minus p times the effect. If I've got no dominance, additive variation is simply the heterozygosity, 2p1 minus p, Hardy-Weinberg, times the squared effect. So loss of heterozygosity means your additive variation is going down over time. In particular, just using that exact same expression, I can write this here. So just as allele frequencies change, your additive variation is also changing. So if I have a finite population size and no mutation, over time I'm going to erode and lose my additive variation and therefore lose the ability for that trait to evolve. Now obviously mutation will come in and we'll talk about that in a second. But this key concept number 2 is drift not only causes allele frequencies to change over time, but it also causes the additive variation in the absence of other forces to become lost over time. Okay. So drift, the power, timescale drift depends upon the population size. Larger populations, more power for drift. Drift has the effect of making heterozygosity go to 0 as alleles become more and more extreme, eventually becoming lost or fixed. That means your additive variation is going to change. The reason we're talking about all this stuff is, what's our big goal? Our big goal is to ask, if I observe a sequence of how means change over time, is that consistent with drift? We have to understand drift to understand that. And the first feature is to ask how additive variation changes under drift. So I keep on mentioning this population size. In reality, there are factors that make the census size different from the effective population size. Let me ask a question. You don't have to raise your hands if you don't want to, but so how many of you have children? So if you look at this group of 50 people, the number of breeding individuals is, let's say, 10. So right away, the genetic size of this population is less than the census size. And that's a very, very common feature. In fact, one of the big findings with genomics is just how much selection has influenced link sites by reducing the effective population size, something called the Hill-Robertson effect we'll talk about in a couple of days. So what I want to know is that, is there some effective population size that sets the strength of drift when the census size isn't a good reflection of how many genes are being passed on? The answer is yes, and there are lots of ways to compute it, and NE replaces N, and there are lots of situations, I'll go through 3, where NE is much less than N. Remember, if NE is small, it means drift acts more quickly. It means you lose variation more quickly, it means allele frequencies fluctuate more, it means selection becomes less effective. So I'm going to consider 3 scenarios: a population bottleneck, unequal sex ratio, and unequal contribution from individuals. All 3 of these scenarios, which are biologically quite reasonable, will result in the effective population size being much less than the actual size. They're what, about, I think it's, is it 6 billion humans now? Thereabouts? If their census size is 6 billion, how come our effective population size is 50,000? Bottlenecks. So when you're dealing with your particular subject and trying to ask about, is the pattern we see consistent with drift, one of the things you need to know is, what's its effective population size? Because you may count 1 million individuals, the effective population size might be 20. Parastrapi, the cabbage butterfly, is a good example. 500 million, couple of billion individuals, easy in the US. If it went through a founding bottleneck, the effective population size may be 20 or 30. You get very different answers comparing it to drift if your NE is 20 or 30 versus your NE is 500 million. So effective population size is important. The bottom line is the effective population size is always, always, always below, and usually dramatically below, your census size. That's one takeaway message. So let's look at varying population size. The key here is the effective population size is highly skewed to the smallest value. Suppose my population sizes were 10,000, 10,000, 10,000, 100. Well, you might imagine, okay, you know, the effective population size is maybe, what, that's 30,000 divided by 4. The actual effective population size is slightly under 400. Suppose you had 1 billion, 1 billion, 1 billion, 100. The effective population size is 400. When you have a bottleneck, this is called the harmonic mean. So what I do is I take the average of 1 over and then take 1 over that. So basically, this thing divided by k is the average of the reciprocals. I take the reciprocal of that, the harmonic mean, that gives me the effective population size. Yeah, question? Sure. Actually, it is exactly captured by this. Let me give an example. Suppose that the cabbage butterfly found it from a single female that came over, and that female was mated to 1 male. So basically, you've got a single, your genetic founding individual is 1 individual. It doesn't matter if they have 100 million adults now, they all trace back to 1 individual. So that bottleneck removes most of the variation. One of the things we'll see, people here, I remember reading the overviews, I thought was someone working on plant domestication? A little bit. So plant domestication is a great example of bottlenecks, where the domestication introduced a bottleneck, and it often makes it difficult to pick out additional signals beyond that bottleneck. So this actually, the harmonic mean actually captures the loss of variation. Now, what you might be getting at are, depending upon what you're looking at, effective population sizes may act over different timescales. If I want to know how many mutations are rising right now, that's a census size. If I observe a certain level of variation in the population, that's a long-term product, and the effective population size there would be the long-term average. So depending upon the question you're looking at, you can actually have different timescales for looking at effective population sizes. If I'm looking at levels of standing variation, it's a long-term historical average. Because the standing variation is drift removing variation, mutation shoveling new variation in. So when you go through a bottleneck, I get this extreme value. Unequal sex ratios. I remember this quite dramatically. My advisor was on sabbatical for a year, which is great. You can put your feet up on the boss's desk. You know, no one else comes around. Feel like you're important. And two people from fisheries, so this is the University of Washington, walked in and said, we're trying to calculate the effective population size of this endangered salmon. Okay. We've got 200 females and got eggs from all of them. What's our effective population size? I said, well, how many males did you use? We used one male. Suppose I've got 2 pollen plants to fertilize 1,000 seeds. This is basically the average. Number of males, number of females, this is how you compute it. If I have 2 pollen parents to fertilize 1,000 seed parents, you might imagine, well, maybe it's 500, half of that. No, it's 8. Whenever you have one sex is more extreme than the other, you again have a bottleneck. Why? Because basically here, everybody traces back to 2 fathers. Not 1,000 fathers, not 500 fathers. So, and these are extreme examples, but remember, if you look at a natural population, you very often have operational sex ratios which are very skewed. You may see equal numbers of males and females, but it may be that 90% of the matings are done by 5% of the males. So in that case, the effective number of males is much smaller than the observed number of males. So again, biology suggests populations have gone through bottlenecks. Biology suggests you probably have a skewed operational sex ratio. Both those things lower effective population sizes. The third thing is unequal contributions. So if, on average, the variance in offspring number is given by sigma squared times m, if offspring are random, it's a Poisson means equal chance, the mean of the Poisson, since I've got 2 offspring, is 2. If I plug in 2 divided by 2, I basically recover n. However, if I've got a variance, so some individuals produce lots of offspring, others produce none, that will again dramatically shrink my effective population sizes. So even just thinking about if 90% of the males don't mate, even if the other males produce the same number of offspring, that gives me a huge variance in here. So variance in number of offspring, which you expect to happen, population bottlenecks, which you expect to happen, and unequal sex ratios, which you expect to happen, this is one way to think about it, are all situations where you basically shrink the effective population size and shrink it very dramatically relative to what you might expect from the census size. So questions so far about drift. The key thing is your population size gets smaller, drift becomes more impactful. It means you lose variation quicker. Losing variation means allele frequencies go to 0 or 1. That means your added variation goes down to 0 very quickly. You need something else to pump in new variation. It also means selection becomes less efficient. If my selection coefficient is I leave 1 extra offspring for every 100, so a 1% selection coefficient, that's a big deal in a big population. If my effective population size is 10, that amount of selection is overpowered by drift. So when drift becomes more impactful, selection becomes less efficient. And we see that in things like codon bias. Okay, yeah,
So
please.
you're talking about rare sex, and I was wondering if that means our sex chromosomes will then have a different.
Yeah, so that's a great point. So sex chromosomes have an effective population size that's about 2/3. Mitochondria effective population size is about, and chloroplast about 1/2. Everything else being equal, they can be even more skewed than that. So different parts, that raises a great question. Different parts of the genome experience different effective population sizes. And I'll talk more about this later, but one of the big findings is, and someone asked about, how much of the variation is structured by neutrality versus selection. The big finding is the neutral theory probably is important because there's so much selection. That sounds counterintuitive, but basically, if I have a neutral allele near a linked site that's under selection, that allele will have a smaller effective population size because selection at that linked site basically makes this variance larger. So if you scan across a genome, what you see are regions where there's low recombination rates have smaller amounts of variation than regions on the same chromosome with higher recombination rates. And the idea is that if I have a low recombination rate, selection to a linked site covers a larger window, and that reduces the effective population sizes over a larger window. We'll talk more about this probably tomorrow. One of the modern ways of looking at genome evolution is something called the Hill-Robertson effect. Bill Hill, Alan Robertson, animal breeders. Selection at a linked site influences nearby sites, and so a lot of the variation in the genome is structured because if you've got sites that are influenced by selection, for example, a gene-rich region or a region of low recombination, those sites have lower effective population sizes. Why? Because past selection, usually to remove deleterious alleles, makes that larger for those regions, and therefore makes the effective population sizes smaller. So one of the key findings from kind of modern genomics is neutral alleles are so common because selection is so pervasive. I have an allele with a fitness advantage here. If it's near a linked site, NE goes down, that fitness advantage may be overpowered by drift. Again, concepts we'll come back and talk about. I have to fight myself to go off on all these massive tangents as we go through the overview, but I'm happy to go on them if people have specific questions who want clarified. So we've talked about drift. So talked about added variation being an important concept. We've talked about drift. And one of the things drift does is removes added variation. Well, obviously something pumps in added variation, and that's mutation. This is the mutational variance. What keeps added variation from being driven to zero by drift? New mutation. Now, if I look at a DNA sequence data, a new mutation would simply be a new SNP or a new indel that arises. But with quantitative trait, and in that case, I'd simply worry about the rate at which that happens. In quantitative traits, the mutation rate by itself is not sufficient. You also have to specify the effect size. So if I have two traits and they both have underlying loci experiencing the same mutation rate, if the effects are much bigger for one trait than the other, I get a much larger mutational variance for that trait. So the quantitative variation is a function of the mutation rate, 2n mu. Mu is the per copy mutation rate. There are 2n diploid copies. And the mutational effects. This is the variation in the effects of new mutations. So again, that looks rather abstract, but it turns out we can estimate this ratio of the mutational variance divided by the total. That's called the mutational heritability. How could we estimate that? It's actually super easy. Take a completely inbred line, brother-sister mating or maybe an inbred plant. Take one individual, blow that individual up, all those individuals are clones, then do a parent-offspring regression over time. That initially is zero because there's no variation. Over time, as mutation accumulates, that parent-offspring regression should become slightly larger and larger and larger. It's a very slow process. You need big sample size, but you can estimate that directly. When that's done, typical values are about 10 to the minus 3. So here is from Mike's and my 1998 book. Here are examples for a whole bunch of traits where they've done these laborious experiments. What you see is these are all about 10 to the minus 3. So over 1,000 generations, you can build up quite a lot of mutational variation. So mutation adds new variation, drift removes it. Obviously, there's some balance in there. And that balance will be important in some of these tests. So drift mutation equilibrium, the simple model called the incremental model, which we'll talk about in a second, gives this equilibrium value. twice the effective population size times the mutational variance. But a really tricky issue is this. So the rate at which mutation occurs is important, but not as important as if this is an allele and it mutates to this allele, what is the model for that? In particular, if this has an effect A beforehand, the incremental model says it mutates to a new allele with effect A prime, which is the current effect plus some random increment. That's not an unreasonable model. If this has a value of, let's say, 1 centimeter of height, a mutation may take 1 centimeter and add on some random value about it. That model is called the incremental model. It's also called the Brownian motion model, for a reason we'll talk about in a little bit. And a lot of people did it. Lynch and Hill, again, Bill Hill of Hill Robertson and Mike Lynch, were the one that did a lot of work on that. This depends upon previous histories, because your new allelic effect is your old effect plus some random contribution to that. That's not an unreasonable model, but the problem is we don't really know what the biology is. So the incremental model, and under the incremental model, by the way, populations continue to diverge over time by Brownian motion. So basically they diverge as a function of time, so you have an infinite divergence over infinite amount of time. There are two other models. Kingman had a very influential model called the House of Cards. For those devotees interested in mutation selection balance on quantitative traits, a lot of work was done by the House of Cards model. And under Kingman's model, your new allelic effect doesn't care what your old allelic effect was. So under the Kingman model, this has an effect of 1 centimeter, let's call it 1 kilo because they're obesity alleles. This has an effect of 2 kilos. Under the Kingman model, when these mutate, both their effects are drawn from the same distribution. They don't care at all about the history. Under the incremental model, you take the 1 kilo and add on some random effect. You take the 2 kilos and add on some random effect. Which model is correct? We don't know. But it matters. The regression model by Xiaobang Zhang and Clark Cockram basically splits the difference. It says I'll take a weight on the current value and then add on a random increment. So if the weight on the current value is 0, I get House of Cards. If the weight is 1, I get the incremental model. And so the bottom line is these two models means that populations don't continue to diverge linearly over time. They converge to some fixed level and stay there. That's exactly correct. There's no effective history. It's gone. That's exactly right. And so the regression model is probably a nice one because it says, well, there's some effect. Here it is. If it's 0, you get, the nice thing about this is a canonical 0 House of Cards, 1 incremental.
But could that be interpreted as phylogenetic inertia?
Well, the reason I'm being hesitant is inertia typically refers to a character. That character, for example, may have a whole bunch of interacting genes in complex pathways with all sorts of levels of buffering. So even though you may change the genotype, the pathway buffering from the multiple genes will constrain it a bit here. This I'm talking about a single allele influencing height. So this may be what underlies inertia, but I don't want to make a one-to-one mapping because there could be other features involved. Your point's well taken. The bottom line is we don't know. And it actually turns out to make a difference. Almost all the analysis is done with the Brownian motion model, where things diverge over time. Under either one of these models, basically you get to an asymptotic divergence. That's exactly what you expect under stabilizing selection. So this is probably a technicality. I'm probably overly worrying about this, but just be aware that's out there. So let's see where we are. We talked about a bunch of stuff. We've talked about added variation, heritability being a central measure for how a trait is going to change over time. We've talked about drift. Drift causes populations, as we'll talk about in a little bit, to diverge apart. It also causes added variation to become lost. We can then shovel in new variation. That's the mutational increment, and they'll give us some sort of balance. And that balance depends upon the effective population size. And that makes sense because if I'm shoveling variation in and I'm draining it, the rate at which I drain the variation depends upon the effective population size. If I drain it slowly, I'm going to have more variation. If I drain it quickly, I'm going to have less variation. See what's next here. Okay. One more slide, then we've done our quick review of quantitative genetics. I should mention here as a cheap advertisement, how many of you know about the Summer Institute of Statistical Genetics in Seattle? Some of you. Have people taken that before? So it's a great thing to take. Did you take the quantitative genetics course when you took it there? Did you take my course when you? My goodness. Okay, I'm feeling. Yeah, okay, yeah. So it's a setting similar to this. In fact, I patterned a lot of this off of here. There are a series of modules which are 10 lectures, 2.5 days. The good news is there's lots of scholarship money. The bad news is the window is almost closed to apply. But there's lots of courses they teach there. So for example, I teach a 2.5-day course on quantitative genetics where we cover a lot of this stuff in detail. We teach a mixed models course. Peter Vischer and Naomi Ray teach an advanced human quantitative genetics course. There are classes on GWAS. So if you're interested in further follow-up courses, Google Summer Institute Statistical Genetics in Seattle because they have a lot of things. And if maybe not this year, think about next year because it's well-funded. There's a lot of support to bring students in for that. National students, international students, is a bit trickier, but still some funds there. So one last thing, and then we'll see if there are questions wrapping up quantitative genetics, is we want to talk about how traits actually respond over time. And the classic expression is due to J. Lush, 1937, called the breeder's equation. And it simply says, what's the evolution? What's the change? Here is a population before selection. Here is the offspring of that population after selection. What's that cross-generation change? R. Well, that equals a within-generation change, S, times the heritability. So if the heritability is small, no matter how much you select, you get very little response. What's this within-generation change? Suppose this is the entire population, and I only let people with glasses reproduce, right? So I take the mean of everyone, then take the mean of people with glasses. The mean of people with glasses is the people that reproduce. That minus the overall mean is the within-generation change. That could be really, really large, but if h squared is small, only a fraction of that gets transmitted. Why? You pick exceptional parents. How good are those exceptional parents at predicting their offspring? If the heritability is high, they're quite good. If the heritability is low, they're not good, right? Heritability is the slope of that parent-offspring regression. If that slope is steep, an exceptional parent is going to have an exceptional offspring. If that slope is very flat, an exceptional parent is going to have an average offspring. So the breeder's equation relates these. There's a lot more. We'll often talk about two different things. The selection intensity, I, is simply S expressed in phenotypic standard deviations. It's a good way to basically ask if you've got corresponding amounts of selection. We'll talk about something, especially the last day, called the selection intensity, which is written here. It looks very similar, and it is in a univariate case. In a multivariate case, I think several people comment on this, the multivariate case, this will allow us to detangle direct and indirect selection. So I can take this breeder's equation here, and I can basically write it, whoops, typo, fix that. I can basically write that equation as this. I write the response equals the gradient times the additive variation. If you've seen this before, this is basically G times beta if you think about the multivariate breeder's equation. So when we talk about how traits respond, the heritability is a central idea. And again, we can write the heritability as the added variation by simply multiplying it by the phenotypic variation. That's your quick super quick overview of quantitative genetics. What are the key concepts moving forward? When I talk about quantitative traits, the phenotype depends upon the genetics and the environment. The genetics depends upon the mating system. If I have clones, my whole genotype gets passed on to the offspring. If I have sexual crosses, only part of the genotype that gets passed on, and the key concept is what gets passed on is the breeding value. The variation in breeding value is given by the added variation. The heritability is the fraction of all variation due to variation in breeding values. If that number is large, the phenotype is a good predictor of the breeding value. If that number is small, the phenotype is a poor predictor of the breeding value. We talked about drift. As your effective population size gets smaller, you lose variation more quickly. That variation can be counterbalanced by mutation pumping in new variation. And we'll talk about how that relates to populations diverging in a second. Then we talked about how traits can change over time. That's kind of the background machinery we're going to need for the rest of the day. We talk about how phenotypes change over time. So any questions about that? How much this was largely reviewed for people? Don't be bashful if it's not, because again, I mean, the population is very divergent. There'll be concepts which will be completely new to you. There'll be concepts you'll be the class expert in. So what I mainly want to do is to make sure that I'm not spending lots of time discussing things that everyone knows, but also I want to make sure I don't skip over stuff that's important. Okay? So questions or comments about our very quick quantitative genetics in, basically an hour?